Hi everyone, my name is TZEPLAEFF, and I would love to present you a project I joined last year when I integrated the Lingo group at the Technical University of Munich. And this project takes part of the maxomod consortium that is funded by Isar Joint Translational Call for Proposal from 2018. The aim of this project is to integrate multiomics profiles and try to reveal early disease signature of ALS. Thank you. So, as you know, ALS is believed to start in the motor cortex ear and over time spread around other cortical areas. So when we study postmortem tissue of the motor cortex ear, we might study like end-stage disease. However, the prefrontal cortex ear only demonstrate intermediate TZEPLAEFF pathology at the stage of death, and therefore we think that studying this tissue might give insight into early ALS disease-mediated pathway. So therefore, we collected the frontal cortex of 50 ALS patients and 50 control patients, but we also collected the prefrontal cortex of transgenic mouse model of the four most known ALS variants, which are C9orf, FUS, SOD1, and TZEPLAEFF. And from the same batch of tissue, we there extracted the RNA, DNA, and proteins, and did a batch of omics analysis. So today I will show you some results of the transcriptomic data, the microomic data, also the proteomics. First, we try to analyze them separately, and finally we try to integrate all of this information to find a pattern like targets for early ALS. So let's start with the result of the transcriptomic in the human patient, and the first step was to determine what would impact the sample. So we wanted to determine if the condition, of course, have an impact, the sex, and also the brain bank origin. And as you can see here, it reveals that the condition only have a few impacts and slightly separate the sample, while we really had like a nicer separation when taking sex into account. And therefore, we decided to really analyze all of our data, male and female, separately. Here's the result of the transcriptomic in blue for male, orange for female, and what you can directly see is that we have much more impact in blue in the male compared to the female human sample. So then, when we wonder which pathway those genes are involved in, here we analyze the KEK pathway of those genes separated by sex, male and female. And interestingly, we do see some pathways that are dysregulated the same in male and female. For example, we have a lot of genes associated with neurodegenerative diseases here, Huntington disease, spinocerebral ataxia. So those genes are activated in both sexes. We also see activation of oxidative phosphorylation in both stress. Also downregulation of pathway associated with immune system response in both stress, and the extracellular matrix is also downregulated in both. But we also interestingly did see some pathways that were sex-specific. For example, in male, we do have specific activation of synaptic-related genes and activation of the ribosomal pathway in female. Then we used those terms and tried with the hierarchical clustering to see in our ALS population if we were able to see subgroup of ALS patients, because we know this disease is really heterogeneous. So we try to better understand what is this heterogeneity. Here, as you can see, we were able to determine 4 main clusters of ALS. Of course, you can always divide and divide, but we say, okay, let's start with 4. And those clusters are called here C1 to C4. And if you wonder what is the first separation that drive the difference, you can see it here. It's mainly driven by gene associated with immune response that is mainly downregulated in the cluster 1 and 2 and upregulated here in red in the cluster 3 and 4. We also observe that cluster 1 and 2 have higher activation of genes associated with synapse, while C3, C4 cluster have higher activation of genes associated with RNA splicing. Then when we further dichotomize our patient, the second separation happens here, and if we wonder what induced this separation, it was mainly associated with gene in the extracellular matrix pathway. So here you can see that in C2 and C4, those genes are mainly downregulated, but they are upregulated in the C1, C3 cluster. So to validate those data, we also did the WDCNA analysis, and we are able to obtain 20 different factors. And how does this work is that it will cluster all the genes that are co-expressed together in different modules. So I gave example of few modules here, and what I think is interesting is to realize that some of the modules really describe better some cluster than others. So for example, if we focus on the turquoise cluster associated with mitochondrial alteration, this is mainly upregulated here in all clusters, C1, C2, and also C4 here in male and female, but it's only downregulated in the C3 cluster. Then when we look at the yellow cluster pathway here, it's mainly associated with synapses, and as you can see, it's upregulated only in C1, C2, but downregulated in C3, C4, exactly what we found in the hierarchical clustering. The TAN module here is associated with immune response, and as you can see, is mainly downregulated in C1, C2, but upregulated in C3, C4, same as in the hierarchical clustering again. And finally, the light cyan here associated with mRNA metabolism and splicing is downregulated in C1, C2, and mainly up in C3, C4. So the conclusion is that molecular differences drive heterogeneity in the prefrontal cortex of our patient, and we are able to subcluster them. So then we focused on the transcriptomic result we obtained on animal model, and here we found very different pathway altered in the different model. For example, C9 was mainly associated with altered immune response. The SOD1 model was associated with change in the MAPK pathway, but also H2O2 response, and the TBP43 was mainly associated with transcription, protein metabolism, and also, I think, you can see it, peptidase activity in both sexes. We didn't see any enriched pathway for FUS. So you might see here, like, okay, it's very different, so what do we learn from it? And I think what is really interesting is when we start correlating all of those data of mouse model with the human sample, and especially with the different cluster 1 to 4. And what we can conclude here is if you look at cluster 1 and 2 of the human sample, it correlates better with the SOD1 model. But when we look at the C4 cluster, then it correlates better with the FUS mouse model. And finally, the C3 cluster then correlate better first with the C9, but also partially with the TDP43 and FUS. So I think this really shows why it has been really complicated lately to validate something in mouse model and then make it work in clinical trials. So we really need to think about which model to use to validate which drug and why we do it. So then we also analyzed microRNA-omic in our human sample. And again, what you can see in blue in male is that we have a bit higher dysregulation compared to female in orange. Then we look at the top 5 microRNA that are dysregulated in our sample, and we focus on this one microRNA 451A because we were able to detect it in, across all our mouse model and also the human sample for male and female. You see it here in big. And what was interesting is that it was mainly downregulated here in female, and we wonder, okay, what are those targets? Why are they all down in female? And here, if you check the targets, what we found is that we have mainly like a protein associated here with the MAPK pathway, but we also have a protein associated with the immune response and the matrix metalloproteins, so really interesting pathway. Subsequently, we also analyzed the proteomic profile of our ALS patients, and again, we did find like much more dysregulation in the male in blue compared to the female in orange. And if you check the terms that are associated with those proteins dysregulated, you again find some similar patterns in female and male. For example, we have extracellular matrix that is altered. We also have the metabolism altered. We have the immune response in blue here and the synapse in orange. But we did also observe like some sex-specific alteration, for example, more gene associated with development in female, transmembrane transport. Interestingly, again, with the proteomic, the MAPK pathway, and in male we had this whole bunch of different terms that are altered but don't really fit together. So it looks like in male it's more broad. So this is really interesting graph. I don't know if you're familiar with, but this is a cluster of semantic space. And technically what it shows is like each round represents here a specific term, so a group of genes that are altered either in the mouse model or in humans. So the color gives you the information of the proportion of those term that is altered in the different mouse model and human. And here you can see that whatever if it's female or male, and I think it's worse for female, we don't really see any overlap between the mouse sample and the human sample, maybe just for a few terms here. But it again shows that it's really complicated to go from mouse to human. So finally, what we try to do once we analyze our omics separately is now, like, what happens if we put everything together? What can we conclude? So we use the MOFA analysis, so it's multi-omics factorial analysis. And again, when we just look at our data, we see a huge separation between male and female. So we decided again to split. And I will not show you everything because there's a lot of graph and everything, but I thought I can show you like an example, like the result we have from the human female results. So in these analyses, we integrated proteomics, microRNA, and transcriptomic. And here what you can see is that we succeed to dissociate 15 different factors. So proteins, RNA that go in the same direction and might work together. And here you have the information of which omic is driving each factor better. And here you have the information of which factor drive the disease information best. So for example here, it's red, so it means it's the factor that explains the disease the most. And when we check, it's factor 12, that is mainly driven by proteomics. So if we check the list of the protein involved in this factor, interestingly, we did in find found some interesting proteins such as NFH. We also see that this factor is driven by the proteoprotein and a bunch of proteins associated in the synaptic compartment. And again here, the MAPK pathway. And therefore, we thought that modulating this pathway might be an auspicious strategy for ALS. So in conclusion, human ALS patients show marked sex-specific differences, and I think this should really be taken into account in future clinical trial and studies. Second, ALS is a molecular heterogeneous. Subgroup can be defined, so I guess like a specific drug would work for a specific cluster of ALS, but not all ALS. Then mouse models should be really carefully identify which model they want to use before starting a study because they recapitulate partially human ALS pathology and correlate with specific subgroup. And finally, the multifactorial data integration highlighted MAPK pathway as auspicious drug target. And if you remember, my colleague Mojan Parva showed yesterday that using FDA-approved inhibitor of the MAP kinase, the trametinib, she was able to restore survival and increase neural length in primary cell culture model of ALS. And also you can, if you go have access to a paper that is on bioRxiv now, you can see that we also use trametinib to restore ALS biomarker in a mouse model. Thank you so much. Just wanted to, yeah, thanks my group, thank you.
Thanks, Laura. The paper is open for discussion. Séverine?
Thanks a lot. Huge database and data. I was wondering, since you showed that you have these clusters with different sets of patients, and you also have the different sex difference, actually, which one is the one that drives the most? Basically, if you would look in each cluster and then look male, female, do you? Basically, because your conclusion is we would have to give different drugs to the different clusters, and basically, would you go by these different clusters, or you already have to give different drugs to male and females, basically?
Yeah. So what I didn't say is that, of course, we decided modulating the MAP kinase was a really nice, maybe opportunity to modulate ALS. And what we observed from the model is that this pathway was mainly dysregulated in the female mouse model and the female ALS. And interestingly, when we validate in vivo in a SOD1 mouse model, we did find restoration in female only. So now we have to determine if it means that it only acts on female and why. Or maybe it's because, as you saw, we always have huge difference in what we see in male and female. Maybe males see more impacts, and then when we treat them with the trametinib, it's already too late. Maybe we have to treat them at a different moment. So we still need to understand if it's like just male and female are different, or we should treat them in a different way.
Yeah, thank you for this very interesting talk. I just have a very naive question, but for human samples, it was post-mortem samples.
Yeah.
But do you know at what stage the patients died? In other words, could some of your clusters be explained by the stage at which the patient died?
Really interesting. I don't have the information of which stage they die, but one of our analysis was to try to see if the different cluster we see, could it be like also like that patient die at different moment in the disease. And indeed we found that, for example, the cluster 4 that was more linked to C9 and high immunity was kind of like showing a late disease of ALS, while the other cluster, for example, the synaptic pathway in cluster 1 and 2 was much likely associated with early disease. And there is a way to see if the genes that are dysregulated can be followed a long time, and it looks like we have some cluster that might represent different stage of the disease.
Okay, thank you.
Thank you. Pietro, and then we.
Hi, I mean, nice talk. So how do you really, one of the issues is always how you differentiate gene dysregulation or cell proportion, right? And is this all cell population driven, or is this really gene dysregulation, or a bit of both?
We check. So what we try also to do is like to determine if what we were seeing were mainly coming from a cell population and another, and I don't think it's really linked to cell population, because in some of our cluster we see like increase of synaptic genes, but we don't have more neurons, So it's like, I guess, like real signal.